Okay, thank you for giving me the opportunity to give you an overview of what is gene therapy, what the basic principle, some clinical results, and some regulatory aspect, I think, which I thought would be useful. And this is my disclosure. So gene therapy is part of the advanced therapies, as you know. So it's based on the EU regulation. It's a way of administering a gene, adding a gene to a cell or to a tissue. And this is can be done, as we'll see, through different routes, through different strategies, or replacing with a new technology a gene, or correcting it more precisely, or even activating another gene, or providing a new gene function. So the two extremes are one side, gene therapy that we do for genetic diseases, where we add a healthy copy of the gene, or the CAR T cells, where we add a gene which is giving a new function to the T cells, which are recognizing the specific antigens. So of course, these are quite complex therapies. They are medicinal drug product by definition, so they are as the other medicinal product. As you will see, it's in parallel for what concerns this audience. Certainly, they go in parallel with the legislation on transplantation, but they are medicinal drug product. They are somatic cells. There is no therapy which is currently allowed in EU through embryonic cells. They are tissue and then there is other type of advanced therapy medicinal product, which are the tissue engineered or combined. So in some cases you will have a pure gene therapy product or a cell and gene therapy product. So the definition is a biological medicinal product that has based on a recombinant nucleic acid, able to regulate, repair, adding or delete a genetic sequence, or it, for therapeutic, prophylactic, or diagnostic effect, relates directly to the recombinant nucleic acid sequence. It does not include vaccines, so gene therapy in terms of EU regulation is not including vaccines against infectious diseases. So how do we deliver the gene to the cells or to a tissue? I'm summarizing here some of the most frequently used vectors, because we've been through this in the past 20 years able to engineer viral vectors. The most used ones for the in vivo approaches are the adeno-associated viruses and also some adenoviral vector, which are DNA viruses. The most used for the ex vivo approaches are the retroviral and in the past 10 years, lentiviral vectors. So depending on the size of the gene that we want to manipulate, that we want to insert, depending on the tropism of the virus, so which cells we want to target, depending on the effect, whether you want to have a permanent effect or a transient effect, whether we need integration or non-integration. These are the tools that we have, which have become quite potent. In addition to that, now, as you may know, we have also genome editing, gene editing technology, which is certainly more precise, because while a viral vector typically delivers a gene randomly or semi-randomly into the genome, through the genome editing technology, we can have nucleases. We have three main types of endonucleases: the Zinfinger, the TALEN, the CRISPR-Cas9, which are able to recognize a specific DNA fragment, a DNA sequence, and they are able to insert and recombinate in this specific point. So this is much more potent technology in principle, because it allows not only to insert a copy of the gene, but it can also disrupt a gene. So for example, if you have a dominant mutation, this can be eliminated. You can have homologous recombination at a specific site. So you can have more precise treatment, avoiding even the risk of insertional mutagenesis, although there is still some off-target site, so some of the points which are not the specific ones. In general, the risk is lower, and also is better in terms of control, of physiological control of the gene that we want to treat. So, as I mentioned, we have mainly two approaches: the ex vivo and in vivo. Certainly, the ex vivo is the one more used in the context of transplantation and cancer therapy and therapy for the some of the blood-borne disorders. The gene, through the gene addition, so the standard vector technology, lentiviral vector, and more recently to the genome editing, there are several disorders which have been treated experimentally. The primary immunodeficiencies have been treated for the past 20 years. Diseases of the platelets, more recently, hemoglobinopathies. There is a great work on sickle cell anemia and thalassemia, and also lysosoma storage disorders, in which actually the gene-corrected cells deliver the enzyme to the different tissues and even the brain. The other approach is the in vivo treatment, where we inject directly the vector, directly the modified virus. So this will transfer the gene into the tissues, whether this is systemically injected, it can be injected locally, such as into the eye, it can be injected into the brain, and this has been used mainly for hemophilia, has been used for some brain disorders due to metabolic problems, neuromuscular disorders. So it certainly needs more the in vivo approach is used for when you need more diffuse administration, or you need to go to some specific tissues. There are advantages and disadvantages of the two strategies. The in vivo, for example, has more risk of immunogenicity because you're injecting basically a modified virus which has an envelope, which has some protein, so you will immediately cause an immune response, which can be controlled through immune suppression, steroid. So there are ways of overcoming this, but this can be a problem. In terms of, I said medicinal product, so most of them are still experimental. I mentioned here the gene therapy medicinal product that received approval in the market, either EU or both, actually, or either EU or United States. And the list is here. These include 2 genetic diseases, such as ADS-KID and Leber amaurosis, and 3 diseases in oncology field, including the most recently CAR T cells. So, what are the real challenges in going in the development of medicinal products which are based on gene therapy? Some of which already I alluded already. The barriers, immune barriers. If you're injecting a virus, you can create immune responses. Of course, if you're having a disease where there is no protein, this is the same as you have for enzyme replacement therapy. So you can create antibodies to the transgene. Some cases we don't know exactly how the disease is working and what is the biology of the cells that we want to target. We need to have adequate preclinical models. We need to have adequate ways of expressing the gene in a right context. And then there are also some safety issues which have been progressively overcome through the improvement of the technology. For example, one is the insertional mutagenesis. If we are used standard vector for delivery of a gene, there is a potential risk that the vector may land in a proto-oncogene, so may activate a gene or inactivate another one. So this has been actually, was a risk that was actually, what actually happened and led to, in 3 type of immunodeficiency, the development of myelodysplasia or leukemias. But now the systems are really improved in the past years. There are also a lot of issues that needed to be solved related to the large-scale production. So initially the experiment was made on small number of cells. If we need to manipulate large millions or billions of cells, we need, of course, to upscale everything, upscale the quality controls, because these are medicinal product require, of course, the quality controls typically of any other drug with specifications which are important for the specific product. And then a number of issues related to the manipulation. You need a special authorization to manipulate this medicinal product because they are made of GMO, so they are genetically modified organism. Specific regulatory needs which were built in the past years, and these are also expensive and need really financial support, and industrial alliances are being key for this. So going now to the most relevant for you type of gene therapy certainly is the therapy based either on hematopoietic stem cells or lymphocytes. The concept is basically the same, so it can be for hematopoietic stem cells an autologous stem cell transplant. The cells are taken for the patients, they are modified, and they are infused. And the genetic modification which uses retroviral lentiviral vector, so which uses vector which integrate, is made through vector that are able to bind to the cell surface of the target cells, release the genetic material, which is essentially RNA for this case, and then this is integrated and then is transmitted to the daughter cells. So in order to be effective for stable efficacy, we need to have integrated vector, integrated transgene, which is transferred to the daughter cells. This is the principle that the different centers are basically applying to the treatment of patients with the stem cell gene therapy approach. So the stem cells are collected from the either bone marrow or mobilized peripheral blood. They are purified through, this is a Clinimax that purified so that we enrich for CD34 cells. And then they are cultivated for 2 to 4 days in the presence of cytokines, in the presence of the viral vector. So this process, which we call transduction, is the time where we infect the cells once from one time to 3 times, depending on the vector, depending on the cells. And in this procedure, the cells get transduced, so they get infected. The vector is transferring the genetic material, and then it ends its life. So the vector was modified to be non-replicative. It loses the replication function. It loses the pathogenetic function because of the dangerous genes have been eliminated. And then the product is reinfused. In some cases, reinfused fresh after the production. In some other cases, is cryopreserved so that all the tests are available at the time of the infusion. And then the patient is followed up. How do we follow? How do we monitor the patients? There are one essential. First parameter is the presence of the vector. So we have measurement of vector copy number, usually, or percentage of transduced cells. And this is done by a molecular test, a quantitative PCR, which basically is a way of measuring the engraftment of the cells and a chimerism. So rather than having a chimerism of donor recipient, we have here a chimerism of corrected and non-corrected cells. And there are specific PCR tests which measure the presence of the vector inside the cells. Then there are 2 usually other parameters which we look, which is safety parameters, the exclusion of replication competent vector. There is a lot of experience now, so this test eventually may be even not considered anymore because there, hundreds, thousands of patients have been infused, and there is no evidence of replication competent retroviral or lentiviral vector. So there is a proposal even to move away from this. And then some test of clonality in order to see whether there is no clonal expansion. So with the current vector generation, it is important to monitor the patient to see whether there is no abnormal expansion. And this is the bone marrow harvest, which is exactly the same. What difference actually is the amount of bone marrow that we harvest is not as much. It was more than one which we harvest from a normal donor because this is an autologous transplantation, and we need the highest as much possible amount of stem cells. So we usually collect 30 to 35 ml per kilogram. And this is the medicinal product that we're infusing. So basically, it comes back from the GMP manufacturing facility, which needs to be authorized for the production of the medicinal product, is a bag and a cell suspension. So the drug is here, experimental drug is a cell suspension of CD34 cells which have been grown and cultured ex vivo. And this is the time of the infusion. In our experience, the facility, the GMP facility is the company called Molmed. This is the example for the beta thalassemia clinical trial. And you see that the number of tests, quality tests that need to be performed in order to release the product, which includes, of course, sterility, presence of endotoxin, immunophenotype, clonogenic progenitors, transduction efficiency, so test of potency, of safety, which are really important to characterize the product. What are the potential advantages and disadvantages of a gene therapy approach for a non-malignant diseases, such as immunodeficiency, for example, and as compared to a allogeneic transplantation? Well, one potential advantage is, of course, this is an autologous transplantation, so the donor is immediately available. We don't need to search it. Of course, in some cases, the autologous stem cells may not be fit for this, for the modification. There are some diseases, as you know, in which there are bone marrow aplasia, myelodysplasia. For example, there is an effort of undergoing effort by Spain for the Fanconi anemia, but the other critical number is the critical parameter is really the number of stem cells which are poor. And of course, you don't want to have too many karyotypic alterations or genetic modification which may increase the risk. The other advantage is that you don't have graft versus host disease, no rejection. The other advantage is the conditioning. You don't really need a full conditioning for autologous gene therapy because it's an autologous transplantation, and depending on the threshold of the disease. So in some diseases, 5% of stem cell engraftment is enough because maybe you have a selective advantage for the lymphocytes. In some other cases, maybe 30%. In other cases, you may need 80%. So you can really tailor and play with the conditioning and combination of chemotherapy. You don't need immunosuppression afterwards. The other advantage is that you can actually put more copies of a gene than a wild type, than a healthy donor. And this is actually the strategy that we have used for one disease, which is metachromatic leukodystrophy, where you want to have high supraphysiological levels of the enzyme. So in this case, we can put more than one copy, and we can achieve more protein. Or you can even use a protein which is modified to be highly effectively translated and processed so that maybe is more active. For example, in hemophilia, now there is a possibility to use a modified factor 9, which is more active than the normal one. The degree of chimerism can be, depending again, 5 to 70, 80%. The highest that we have seen in our clinical trials is 80%. So we have not seen really full chimerism, but probably we are there. The other potential advantage is that now we have started from severe disorders. We are moving progressively to less severe disorders. So that's the potential that once the safety is well established and there is long-term observation, autologous transplantation is easier, more suitable than an allogeneic transplantation. So this potentially could be applicable also to less severe forms of the disorders. And then oncogenesis, which is, as I mentioned, is a safety risk potential, which has not been seen with the self-inactivating vectors, the retroviral and the lentiviral vector of the most recent generation. In terms of ex vivo gene therapy, we know that the source is important. As I mentioned already, mobilized peripheral blood and bone marrow are key for a key cell source for the gene therapy approach. in the future, we have heard already today some interesting presentation about the use of alternative conditioning regimen, which may be used in alternative to chemotherapy, so like of antibodies. I think I already did this. And this is an example about the chemotherapy, the conditioning, where we, in our clinical trials, we have changed, depending on the disease, the type of conditioning with low intensity with the ADS-KID and higher intensity with, for example, with beta thalassemia. And for the metachromatic leukodystrophy, where we want to target also the resident myeloid cells, we use busulfan, which facilitate the crossing of the blood-brain barrier. So the ex vivo autologous stem cell gene therapy is being used for the immunodeficiency, for lysosomal disorders. The principle is that when you do the manipulation of the cells, the ex vivo manipulation, really you need to preserve the stem cells because you need to maintain their self-renewal capacity, their capacity to engraft and to generate all the different lineages. This primary immunodeficiency, in particular combined immunodeficiency, were the first one to be treated with gene therapy. The first clinical trial started in the 90s. The first successful one actually near 20 years ago. Our experience in ADS-KID, which is disorders caused by the accumulation of toxic metabolites, was successful, and we published some time ago the results of the first 18 patients who were treated with autologous stem cell gene therapy, showing 100% survival, about 80% intervention-free survival, a reduction of severe infections, and correction of the immune and metabolic defect. So these data were then used by the company GSK to obtain the marketing authorization in Europe for the gene therapy product. And since then, 5 patients have been treated with the commercial product, which is called Trinvelis. In addition to the ADS-KID, lentiviral vector have been used more recently in the past 10 years, and this is because they alleviated the risk of oncogenesis and are highly effective in transducing stem cells. So they're based on HIV vector, which 90% of the genome is eliminated during vector construction, and it leaves only few regions which are the ones necessary for the cell infection and basically for the copying of the cDNA, of the RNA into cDNA, but these are given in a way that they are not transferred to the cells. So they are basically used to build up the virion, the particles, but the genome of the HIV is essentially nearly all cleared by the pathogenetic proteins. The experience that we had with HIV is with different diseases. I just want to give you the example of this lysosomal storage disorder, which is metachromatic leukodystrophy, which is caused by an accumulation of the enzyme by toxic metabolites, sulfatides, in the central nervous system and in the peripheral nervous systems due to the lack of the aryl sulfatase A. This is a very severe disorders with children who are born and have no treatment available. We know that allogeneic transplantation can be effective, but only in the forms with later onset. And so the goal here was to really exploit the gene therapy and the progeny of the autologous stem cells to make them delivery to the differentiated tissue, and particularly to the brain, the missing enzyme, the aryl sulfatase A, so that the enzyme could be then uptaken by the neural cells by the oligodendrocytes, and these basically these myelocytes, the monocytes, macrophages, go into the brain, could produce high levels of the enzyme, which could be uptaken and then have a therapeutic benefit. This concept, which was developed by Dr. Biffi and Dr. Naldini at our institute in Milano, eventually turned to be correct. The hypothesis turned to be correct because when we measure the presence of the transduced cells, and this is done by analyzing the single progenitor colonies, we have this first patient, we have 20 to 80% of the stem progenitor cells which are corrected. They produce high levels of the enzyme in the blood, but most importantly, they also produce normal levels of the enzyme in the CSF. So suggesting that we have achieved local administration of the enzyme through the blood cells which migrated there. And this results in a dramatic change in the outcome of the disease. Just to give you two examples, this is a score of the magnetic resonance, the MR score, which the higher the worse, and this is the score of the patient treated with gene therapy, which differs greatly from the patients who were of the natural history study. And also the motor function. These patients progressively, the natural history are here, down here. They not able to acquire a motor milestone, and they are regressing, and they progressively lose the capacity to move, to speak, and to eat, and eventually they would die. These patients, the treated patients with gene therapy, are acquiring a normal or near to normal motor function and also cognitive function. However, we will learn from the experience of these patients that we need to intervene early, so that we need to intervene really in a pre-symptomatic stage in the children who are born in the families where there is already an affected child. I think I'll give you just brief example of other disorders which have been treated with gene therapy, non-hematopoietic cells, but the epidermolysis bullosa is a severe skin disorders, and here the cells are taken from the skin for the patients. They are corrected ex vivo, again in the lab. You see these are the keratinocytes which are corrected. This is a disease which is a devastating disorder because basically the skin detaches and forms bubbles because of the lack of an integrin, of an adhesion molecules. And the children and the patients are implanted with the autologous skin, which is genetically modified to express the missing protein. And this is a recent publication by the group of the Dr. De Luca and an Austrian group who showed that 80% of the skin of this child basically was restored, and the child now is doing fine, and he was in a really life-threatening condition, like a burned child. Another approach which has been used successfully for the in vivo gene therapy is the gene therapy for FARTO9. Several papers came out and really show now that we can reduce up to basically zero the frequency of the event, of the bleeding event in a patient with hemophilia, and reduce the use of clotting factor. Also very impressive results which came from the recent publication on spinal muscular atrophy using viral vector delivering the missing gene. It's a specific subtype which is called AA9, which probably is going to the brain, although the mechanism is not yet known. And then there is the whole the oncology field. I certainly not going to go into much detail because there are many presentations about these topics, which is very hot and impressive. But the principle here is that we can take the T cells of a donor or maybe of a patient, and use viral mediated or even potentially genome editing to introduce new receptors to modify the specificity of a T cells, such as the CAR, such as the TCR, different approaches are there, and this has been used successfully for the leukemias and lymphomas. There are a lot of groups working now on solid tumors. We can also modify the cells to include the suicide genes, for example, in the context of allogeneic transplantation, using TK suicide gene in order to control immune responses and graft-versus-host diseases in case the graft-versus-host diseases manifest. We can modify the homing of the cells, facilitate the homing to specific organs, increasing the function by potentiating them, for example, with cytokine production, or modifying the resistance to some cancer mechanism, and so on. So there is a great potential, and the field is really expanding. Again, these are gene therapy products. In some cases, they consider cell therapeutic product with a modified gene, but usually this gene therapy product. And again, they follow the rules of gene therapy. And the example of the anti-CAR, of the CAR anti-CD19 T cells, I'm sure you're well aware of the response that was seen in these patients with acute lymphoblastic leukemia by different groups, and this is just two of the papers, but there are of course several groups in Europe which are working on this. One important aspect is the safety, and indeed we've heard about potential neurological toxicity, the cytokine release syndrome, risk of allergy. So while we increase the potency, we also have to keep an eye on the safety, and now there are measures to overcome in part the risk of hyperactivating the immune system and the risk of unknown adverse event. One, of course, is the fact that if we use an antigen which is directed to specific autoantigens which are present also in normal tissues, we may end up in having toxicity in the brain, in the heart, in organs which carry also that antigen. So the more we have a potent tool to destroy the leukemic or the tumor cells, the more we have to be careful that this is specific for the tumor as possible. And then there are also ways of now modifying and expanding T cells, maintaining their capacity to differentiate into different T mature cells, in expanding and maintaining the stem cell, again through viral vector and genome editing. Genome editing is already mentioned, is the principles. They have three tools which have been used in preclinical studies. There are clinical studies which have just started for a few diseases, so we'll see how these tools will behave in the context of clinical trials. And I think I will okay, so I have just a couple of minutes, and then we leave for some questions, because you can find them in the slides. But the main concept that I wanted to say is that you have to give the gene therapy field is within the medicinal product field. It has to follow a number of regulations, which I'm not going to detail, advanced therapy. But basically, I'm summarized here the fact that when you have to deal with a gene therapy product, which is a stem cell or lymphocytes, of course, we have to go in parallel with all the other regulation, which is related to the allogenic cell transplantation. And so it's quite a complex field, because you have to follow both in parallel the gene therapy regulation and also the tissue-directed and cell-directed, because you need the traceability. You need, for example, to make sure that the donor has no infection. There are some type of infection which, as you know, would disqualify the donor. There are ways of overcoming this, for example, by manipulating the cells in a specific environment. So I think it is important when establishing a gene therapy program to look at all these aspects. And the final other important information is that according to the current EU law, a medicinal product is approved and goes to the market through the centralized procedure. So in the EU, it has to go through the centralized procedure in the evaluated usually by a specific committee of the EU, which is the CAT and also the CHMP. Okay, I'll stop here and thank you for your attention.